Welcome to Introduction to Ecology, Lesson 15. In the last class, we talked about predation. Today, we're going to focus in on a very specific type of predation, and that is parasitism. So a couple of definitions before we start. A symbiont is any organism that lives in or on another organism. A parasite is specifically a predator that lives in physical or physiological contact with its prey, the host, and consumes certain tissues or bodily fluids. Now, like we learned in the last lecture, parasites typically harm but do not immediately kill the organisms they eat. A pathogen is an organism that causes disease. And so a good example is malaria. Malaria is a symbiont to the extent that it lives in or on other organisms. It lives in or on people. It's also a parasite because it lives in physiological contact with the human, in human cells, and it actually consumes part of the human tissue to grow, part of the red blood cells. It's also a pathogen because, as most of you know, malaria causes disease. It causes one of the largest burdens of disease across humans worldwide. Now, malaria is a parasite that is a pathogen, but not all parasites are pathogens. In fact, the negative effects of parasitism vary widely. So you can have some parasites that have a very mild negative effect on people or on other animals. A good example in people, of course, is pubic lice. So some of you may or may not have these, but they're probably not causing you tremendous amounts of damage. They're unpleasant. They're not great. It's best to not have these, but again, no one's going to be very, very sick from pubic lice. Different kind of story with what we call trypanosomes or Chagas disease. So this is a parasite that lives in you. It's spread by kissing bugs or triatomine bugs, and it actually damages your heart. And so if you have Chagas, frequently you don't know you have it. You pick it up when a bug bites you in the middle of the night, and then years later you end up with heart failure because of the damage that this particular parasite has done. So this parasite lives in you for many, many years before it kills you, but ultimately it does kill a fraction of the population. One interesting thing about parasitism is that it can impact host behavior, and typically it does this to aid its own spread. So a common example, one that people in biology study, is Toxoplasma gondii. This is a microscopic parasite, and it has to complete part of its life cycle in cats. But other parts of its life cycle take place in other animals, and a common animal that another part of its life cycle takes place in is rodents. And so this parasite actually causes rodents to run towards cats. And as a result, the rodents are more likely to be eaten by the cat. And as a result, the parasite can get into the cat body, and it needs to be in a cat to complete one portion of its life cycle. Another good example is a worm that's actually found in ants. This is a nematode or a worm that turns the butts of the ants red. We call it berry butt. And it actually causes the ants' behavior to change and causes them to climb high up in the trees. Why does it do that? Well, what happens then is that the ants, because they look like berries in the tops of trees, get eaten by birds, and that's what the parasite needs for transmission. The parasite needs to be in the ant, but then it needs to be eaten by the bird and moved to another location where there's more ants. All right, so sometimes we think of parasites, we think of things like malaria, we think of toxoplasma, and those are little. But in reality, parasites vary greatly in size. They're not just tiny things that we can't see. So we actually have what we call macroparasites, and these can be seen frequently with the naked eye. Macroparasites are arthropods, so things like insects, things with crunchy skeletons or exoskeletons, and worms, nematodes, things like that. So here's an example in a fish. This is a parasite that you can see. It's actually as big as the fish's tongue. And what it does is it eats the fish's tongue and then it replaces the fish's tongue. And it lives there inside the fish's mouth, sort of getting the food that the fish takes in. But clearly that's visible to the naked eye. We do have, though, microparasites, and these are parasites that can't be seen with a naked eye. Typically, these are things like bacteria, protists, unicellular fungi. And so here's an example is leprosy. Leprosy, we cannot see individual leprosy organisms. We can see their effect on people. We can see sort of the blackening of the skin and the leprosy on people's hands or bodies. Not as common anymore, but very common in biblical times. But we can't see the individual organisms. All right. Most host species, including people, are attacked by parasites. And in fact, the vast majority of host species are attacked by more than one parasite. And so you can see different types of organisms, how many parasites on average they have per species. So fish don't have too many. Mammals have quite a lot. Depending on the type of insect, butterflies and moths have a lot of parasites. Beetles have fewer. And then trees. Trees have parasites numbers that are off the chart. So many, many different species of parasites attack each type of tree. Now, most parasites, by contrast, only infect one or a small number of hosts. So although most hosts are infected by tons of different parasites, each individual parasite tends to be very specific to one or a few hosts. And that's because the parasites need to be really closely adapted to the hosts in order to sort of sneak around all the defenses that the host has. We'll talk about that in a little bit. But it really takes being specialized to be able to get into a host or get onto a host and manage to eke out a life there. Because most hosts are infected by at least one parasite, and because frequently those parasites are specialized for that individual host, parasite diversity is incredibly high. In fact, some people estimate that 50% of all species on Earth are parasites. And again, that's because most host species have parasites. So for every individual host, we have at least one unique parasite, or most of them have one unique parasite, and most of them actually have multiple unique parasites, or at least multiple parasites that are specialists on them and a few other types of species. Like everything else in the world, parasite diversity varies as you change across the globe. And so here we have an example of global primate diversity, primate parasite diversity. And what you'll see is that it tends to be very high in the tropics, where primates are much more abundant. All right. Very broadly, we can divide parasites into two classes. We can divide them into different types of classes. We already saw dividing them into macroparasites and microparasites. Another way that we can divide them is into ectoparasites and endoparasites. So ectoparasites are parasites that live on the outer body surface of its host. So for plants can be parasitized by ectoparasites. Plants can be ectoparasites. One good example is what we call holoparasitic plants. These are actually plants that cannot photosynthesize. So they get their water, nutrients, and all of their carbohydrates by living on the outside body of a host. Sometimes that's a plant, sometimes it's a fungus, and then they suck all of that they need out of the host. So this is a good example. These are actually plants you've probably seen if you've been out in the experimental forest. They're quite common. You might have thought that they were fungi because they weren't green. They aren't green because they don't have any chlorophyll, so they can't make their own food from sunlight. In fact, they derive their food from the other host plant that they live on. We can also have hemiparasitic plants. These are plants that can photosynthesize, but they still can't do everything for themselves. So they need to get water and minerals from their host, but they can make at least some of their own carbohydrates because they have some chlorophyll. So again, hemiparasitic plants are going to look a little green, but they can't sort of get everything they need from abiotic sources. and so they actually pull them from biotic sources. They pull them from the host plant or fungus that they live on. Fungi can also be ectoparasites. They can be ectoparasites on plants, so things like rusts, you see these on the outside surface of the plant, and so that means that they are ectoparasites. Smut is another thing. Smut infects a lot of crops, a lot of plants that we eat, and again, it's a fungi that grows on the outside surface of the plant. Mildew, another plant ectoparasite. Again, we know it's an ectoparasite because we see it on the surface. And mold. Mold grows all over plants. It grows all over a lot of things, sometimes the fruiting bodies of the plants. And again, it's an ectoparasite because we see it on the surface. We also have ectoparasites that are fungal that live on people. And a great example is athlete's foot. Athlete's foot actually lives on the surface of your skin, typically in the foot area because that's where the habitat's right for it. And it causes us to itch and it's unpleasant and we try and get rid of it. Animals can also be ectoparasites. So if I think about a plant tends to be lived on by things like aphids and whiteflies and scale insects. We talked about these in the last class, or at least scale insects in the last class. These are insects that sort of live on the plant. They tend to live on one plant their entire life, and they basically suck plant juices out. And so these are ectoparasites. They're on the outside and they're living on the plant. If we look down to the roots, there's also ectoparasites that live down there. Some of them are insects, things like beetle larvae, cicada larvae, but we also have nematodes, little worms that live on the plant roots and that suck the juices out of the plant roots. And of course, just like plants, animals can be parasitized by other animals. They can have ectoparasites that are other animals, and a good example is fleas. So a dog might have some fleas that are living on its body, and those fleas are basically drinking its blood. Well, those fleas are ectoparasites. And of course, humans can have things like fleas too, or lice. We can have pubic lice, like we talked about before. That's ectoparasites. You can see those. And we can also have lice in our hair. Nowadays, that's coming back. That was a thing sort of back many years ago. People got rid of them, but now the fleas have developed resistance, or the lice have developed resistance, so kids in school these days have to worry about hair lice. All right, so that's ectoparasites. We also have endoparasites. Endoparasites actually live inside their host. So they can live in any tissue in the host. They can actually live inside host cells, so not just sort of in the spaces in between the cells in the tissue. They can actually live inside the cells, and they can live in the alimentary canal. So the alimentary canal is a tube that goes from the mouth to the anus. We have one, other animals have one, and it's a great habitat for endoparasites. Why is it a great habitat? Well, it's basically this opening to constant food source. We are constantly putting food into our alimentary canal in our mouth, and then we process it through and it gets excreted at the anus. So, not surprisingly, many of the parasites that live in the alimentary canal don't actually eat host tissue. They're not actually eating your stomach lining, or they're not actually eating your throat lining or your mouth. Instead, they're eating the incoming nutrients. We still call them parasites, even though they're not eating your tissue, because they're living in close proximity and eating what will eventually become your tissue. So we still call them parasites. But again, the ones that live in the alimentary canal don't tend to eat you, they tend to eat the food you're putting in. Great example is a tapeworm. So a tapeworm actually uses what we call a scolex, which is a structure with suckers, sometimes hooks, to attach to your alimentary canal. So one problem with the alimentary canal is that we're putting food in and it's constantly moving through. We put it in the mouth, it moves through to the anus, and that creates this flow. We actually have peristaltic motion that pushes that food down through the alimentary canal. So it can be tough for the parasite, the endoparasite, to stay in the alimentary canal. It might get excreted in the poop, and sometimes it does, but a lot of endoparasites that live in the alimentary canal, they have ways of staying in there. And so with these tapeworms, they use this scolex to attach to the wall, and that allows them to stay there even though stuff's moving through, And as they hang out there, they absorb the food that the host's already digested. They sort of absorb the food that your stomach has already put acid into and broken down, and that's a great environment for them. So like I said, some endoparasites live in the alimentary canal, but other ones actually live in body tissue or actually inside of your cells. So a great example is chlamydia. Chlamydia is a bacteria that lives inside human cells. It's an intracellular parasite. And so they have to get into the host cell. And once they do that, they actually use the energy and nutrients from the host cell to divide and replicate and make more chlamydia bacteria. Just like animals, plants can also be affected by endoparasites. One example is bacterial soft rot. So you know, as compared to smut or rust or mold or mildew, with soft rot, you don't see anything on the surface. The plant kind of degrades. It just kind of wilts up. That's because there are internal parasites living inside the plant that are causing it to sort of decay and liquefy. All right. So again, we have these two broad strategies of parasitism. We have ectoparasitism and endoparasitism, and both have advantages and disadvantages. So if you're an ectoparasite, one of the advantages is that it's pretty easy to disperse, right? You're on the outside. You don't need to get out. You're already on the outside. So if I'm a flea living on a dog, all I have to do to disperse is kind of hop off. Maybe the dog brushes against another dog and I switch to a new host. So that's pretty easy. If I'm an endoparasite and I'm living in a gut, or I'm living inside cells, or I'm living inside other tissues, it's harder to disperse to a new host. So, you know, if you're killing your host, you got to get away, and that can be quite difficult. So endoparasites tend to have to have specialized strategies for moving to a new host. Ectoparasites, another advantage of being an ectoparasite is that you're safe from the host immune system. Typically, the host immune system has trouble getting at you when you're on the outside surface of the body. Now, the host does maybe have some defenses that it can use. Say you're sucking blood, maybe the immune system has some chemicals it can sort of produce that help to try and deal with these ectoparasites, but it's not as effective as dealing with them when they're actually inside the host body. Endoparasites, because they're actually inside the host body and frequently inside host cells, they're very vulnerable to the host immune system. The host immune system is sort of constantly patrolling the tissues and the gut and the inner parts of the body of the host, or the inner parts of the body of the plant, and so it's much more difficult for endoparasitisms because they have to deal with this line of defense. On the other hand, endoparasites are actually safer from natural enemies. If you're an ectoparasite living on the surface of an animal, nothing's protecting you from the other stuff around you that might pick you off and eat you. So ectoparasites can actually be picked off by the host and eaten. But that's a lot more difficult for endoparasites because they're inside the host. So yes, they have to deal with the immune system, but they don't have to deal with other things that are trying to get them from the outside. Same kind of deal, endoparasites have the advantage of being protected from the external environment. The host's body is typically a sheltered environment. Frequently it's held at a constant temperature, which can be an advantage. Frequently it's held at a constant moisture rate. Even if it's something like a plant that's not held at a constant temperature, or an ectotherm that's not held at a constant temperature, it's still a protected environment. There's still less wind, there's less rain, there's less sort of external factors that can be very hard to deal with. Another benefit of endoparasitism is that it's pretty easy to feed. It's much easier to feed as an endoparasite than an ectoparasite. As an ectoparasite, you've got to get through the host's outer surface and, got to get through that in order to get the good stuff inside. If you're an endoparasite, you're already inside. You're already in there with the good stuff. And so it's pretty easy to feed. You might just hang out in the host gut and absorb the nice pre-digested food as it passes by. That's pretty easy. So like I said, parasites that are ectoparasites, one of the big advantages of being an ectoparasite is that it's pretty easy to disperse. you just kind of hop off the host and hop onto a new host. Sometimes you do that through the environment, sometimes you do that directly when one host touches the other. So for example, with pubic lice, you can do that when one host touches the other, or you can do that through the toilet seat. Both options are available to you. Now with endoparasites, it's a bit harder. With endoparasites, you're inside the host, so you got to get outside and then you got to get to a new host. You got to find that new host. So there's a number of different ways that endoparasites can do this. One of them is to alter the physiology or behavior of the host. And in fact, we saw that with toxoplasma, where the toxoplasma causes the rodent to run towards the cat. Well, that's because the toxoplasma needs to find a new host, and so it needs to get into the cat. Another way that parasites that are endoparasites have evolved to disperse is by developing these complex life cycles with stages that are specialized for dispersal. And there on the right, we actually see rat lungworm life cycle. And this is a little bit like toxoplasma in that it needs to complete its life in different hosts. It spends some of it in, the larvae mature and lay eggs and develop their first stage in the lungs of the rat, hence the word, but then they're actually excreted into feces and they're eaten by mollusks, and then the mollusks are then eaten by the rats, and around we go, and actually when humans eat those, the humans can get very, very sick from rat lungworm. So again, this is sort of a complex life cycle, but we actually have a very complex life cycle when we look at malaria. Malaria is another endoparasite. Like we saw in the first slide, malaria actually lives inside the red blood cells of the human or primate host. So how does it get from one host to another? Well, we probably all know that it uses a vector. It uses mosquitoes to sort of get taken up and then moved into a new primate or human host. But to do that, malaria needs to have a complex life cycle. It needs to have different stages that are adapted to live in the human host and other stages that are adapted to live in the mosquito host and then sort of to be transmitted from one human host to another. So the sporozoite is actually what we find in the mosquito salivary glands. This is the form of malaria that the mosquitoes have when they're infected and that they actually inject into the human during a bite. But once the sporozoite gets into the human, it doesn't stay as a sporozoite. What it does is it travels down to the liver and it divides to form what we call a merozoite. A merozoite is a different, a very like physically it looks different form of malaria, and that's what we get in the liver. Now within the liver, what happens is within the liver and within the red blood cells, some merozoites replicate and produce new merozoites, but other merozoites, as they replicate, they actually turn into what we call gamete-producing cells. And gamete-producing cells have male gamete-producing cells and female gamete-producing cells that produce male and female gametes. So these basically have sexes. Believe it or not, malaria has sexes. And these two different male and female gametocytes, these gametes get taken up by the mosquito during a blood meal. And when mosquitoes take in male and female gametes, the male and female gametes can actually fertilize one another, and that produces zygotes. So the zygotes form in the mosquitoes, and then the zygotes turn into sporozoites, and that completes the entire cycle. So again, a very complex life cycle with all these different stages, with the sporozoite stage, the merozoite stage, the gamete-producing cells, the gametes themselves, and then those gametes fertilizing to form zygotes. And the different stages occur in the different hosts, the cycle in the humans versus the cycle in mosquitoes. All right. Not surprising, just like with predation in general, parasites have ways of getting into hosts, and hosts have ways of protecting themselves against parasites. So let's take a look at how hosts protect themselves against parasites. One good advantage is just to have a protective outer covering. So a lot of organisms have protective coverings that keep ectoparasites from piercing their body or even attaching. A lot of plants have sort of thick woody tissue that prevents ectoparasites from piercing through to the good plant juices inside. Protective outer coverings, just physical barriers, can actually make it difficult for endoparasites to gain entry too. One of the reasons we have skin is because skin forms a protective outer layer that stops endoparasites from getting into our bodies. Skin is very good at sort of getting rid of these stuff that's trying to get inside of us. And in fact, skin sloughs off every couple of days, and that's a way of sort of moving the stuff that's trying to get in out, getting rid of it. Hosts also have immune systems. So vertebrates have very complex immune systems that involve different types of cells that attack endoparasites. Remember, immune systems are primarily good at attacking endoparasites. They can do a little bit for ectoparasites, but not much. They're really good at attacking endoparasites. And so the vertebrate immune system has multiple different types of cells. We have memory cells that actually recognize things that have attacked us in the past and are primed and ready to go to get rid of those in the future. We also have just generalist cells that swim around our body and find things that don't look like our own cells and attack them and engulf them and destroy them and eat them up. We also have types of cells that spit out antibodies, little markers that tag invaders as sort of something that needs to be destroyed, and then we have other cells that come along and destroy those. So you do not need to know, this is not a course in immunology, you do not need to know all of those individual T1, Th1, Th2, macrophage, killer cells, B cells, T cells, NK cells. You don't need to know those. You do need to know the different strategies that the immune system has in vertebrates for destroying invaders, but you don't need to know the specific types of cells that do that. That's just a figure for your benefit. So plants actually have immune systems as well. They don't function the same as human immune systems. They don't have those T cells and B cells. They have a number of different defenses, the different immune system defenses. So one is resistance genes. And these are genes specifically coding for proteins that can sort of lock onto and recognize different invaders, different parasites. We'll talk about that in a minute. They also produce antimicrobial compounds. So this is sort of a strategy for killing specific microbes that are common attackers. Plants will also produce chemical warning signals that sort of protect other neighboring plants. And they will also, when they are sort of attacked by a parasite, that will stimulate lignin production. And lignin is basically this tough material that they upregulate lignin production, and that produces a barrier on the external part of the plant that will protect them from the parasite. So going back to resistance genes are really interesting in plants. So the way resistance genes work, they're proteins that are the exact right shape to lock on to sort of the shape of the invading parasite. Frequently it's a bacteria. And so they'll perfectly match that shape like a puzzle piece, and that will provide resistance because the part they lock onto will be the part of that parasite that the parasite needs to attack the plant. And so that prevents the parasite from attacking the plant. Now, the interesting thing though is that the plant tends to have many different resistance proteins, but each resistance protein only locks onto one particular parasite. It only locks onto a parasite of exactly the right shape. And so you can see that in that figure down on the bottom, we have in the top left, we have the big R protein perfectly matching the big A protein on the bacterium. The effector protein on the bacterium, we call it an effector protein on the bacterium, or an A virulence protein on the bacterium. Those are the same thing, effector and A virulence proteins. We have that, and then we have the resistance protein on the plant. And the resistance protein, big R, perfectly matches the effector or A virulence protein, big A, and so that provides protection against big A. But the resistance protein, little r, does not perfectly match the effector protein, big A, and so it does not provide protection for that. And so in that top right corner, you see we have disease because we have the little r trying to protect against the big A, and it just doesn't match. The big R does not match the little a effector protein. The big R resistance protein does not match the little a effector protein. And so again, in that bottom left corner, you can see that we have disease because the big R resistance protein only matches the big A effector protein, not the little a effector protein. And so it can't protect against the little a bacterium, and so we would get disease. Same thing, that little r protein does not match the little a effector protein. The little r resistance protein does not match the little a effector protein, and so in that bottom right corner, again, we have disease because it's just not a perfect match. You have to have a perfect match where that resistance protein is exactly the right shape to lock on to the effector or avirulence protein and protect the plant. So when this occurs, when we have one gene protecting against one bacterium, we call that a gene-for-gene interaction. So that means a particular plant genotype provides resistance to a particular parasite genotype. And so this is an interesting scenario because bacteria tend to evolve new effector proteins in response to the selective pressure from plants being resistant from the resistance protein. And so we get new alleles arising through mutation and then spreading through the population of the parasite in response to the selective pressure from the plant resistance gene. All right, so hosts also have ways to protect against, another way that hosts can protect against parasites is through biochemical defenses. So bacterial and fungal endoparasites tend to need iron to complete their life cycle. And so what we and other vertebrate hosts often do is we rapidly deplete iron from our blood, and that basically starves the fungi or bacteria of the iron, and this can actually cause them to be kicked out. We do this using an enzyme known as transferrin. It actually just sequesters the iron and makes it unavailable to the parasites. That's something that vertebrates do. Plants tend to secrete secondary compounds. Again, secondary compounds were those chemicals that the plant makes that are not part of its body tissue. They're not the primary compounds, but they protect, and secondary compounds can protect against the big predators like herbivores, but they can also be produced to protect against things like bacteria and fungi and other parasites. Animals will often eat plants that have secondary compounds to treat their own parasites. Often these secondary compounds, as opposed to resistance genes, secondary compounds can have quite broad effects. They can target a lot of different types of bacteria, one single secondary compound, and so often they're effective against the parasites, the fungal or the bacteria parasites that are in animals as well, and so animals will eat them as a way of combating their own parasites. And so this is what leads to medicinal plants. That's us using the secondary compounds in plants to sort of kill the parasites in our own bodies. Another way that hosts can defend themselves is through the use of defensive symbionts. This is where we actually have mutualistic symbionts, things like other bacteria and fungi, that help to protect the host against parasites. And they do that in a number of ways. They can do that by competing with the parasite, and so they sort of take up space or take up resources that the parasite would otherwise need or needs, and so they outcompete the parasite, and so the parasite can't live on the host. They can also do it by stimulating the host's immune system and causing the host's immune system to be activated and primed, not enough to kick out the symbiont, the mutualistic symbiont, but enough to kick out the parasite. So this is a figure from 2018 paper, and it shows the different places where defensive symbionts exist, and so you can find them on the outer surface, the integument of animals. You can find them more internal in the guts. You can actually find them within the body tissue, or actually you can find defensive symbionts inside cells of the host. And depending on the particular symbiont, they can be acquired horizontally, what we call horizontally, or vertically. It means they can be acquired almost entirely from the environment or from contact with other members of the species. And we frequently find that with stuff that's more on the outside. Again, that's because, these things on the host surface, it's easier for them to disperse from one host to another, or from one host to the environment to another host. We also have what we call vertical transmission. That's where it's passed down from the mother, or passed down from a parent, usually the mother. And that we can find that with stuff that's on the outer surface, on the integument of the animals. But we can also, that's the primary way that these more internal mutualistic symbionts are passed down, things that are inside the cell or inside body tissues. All right, not surprisingly, just as hosts have ways to defend against parasites, parasites have ways of getting around those defenses and getting into hosts. They have to, or else they can't survive. So ectoparasites face similar challenges to what we saw with herbivores and carnivores. They're trying to deal with hanging out on the outside of the host and kind of dealing with what the host is doing to try and get rid of them. So again, last class we talked about how some insects in particular will detoxify toxins that a plant produces. Something like an aphid on the right, feeding. Often it has ways of either ignoring or getting rid of the toxic chemicals the plant is producing to try and stop it from feeding. Also, the aphid has a stylet that is sort of a needle-like mandible. It can stick into the host, and that allows it to deal with the structural barrier of just that plant tissue being there, and it needs to get in through that tough, leathery plant tissue to get at the plant juices. On the left-hand side, you see chimpanzees, and they're busy pulling off ectoparasites that are living on each other, and they frequently eat those ectoparasites. And so a lot of ectoparasites will sort of camouflage themselves or burrow into the hair and be harder to find, and that's a way of getting around that behavioral adaptation that the hosts have. It's a behavioral adaptation on the part of the parasites for getting around the behavioral adaptation of the host, where the host is looking for them and pulling them off. Endoparasites, it's a different story. They face unique challenges. They face the host immune system, which is actively trying to get rid of them, and they face host biochemistry, things like the transferrin enzymes that are busy sort of altering the host chemistry, getting rid of things like iron that they need and trying to starve them out or get rid of them in other ways. So encapsulation is actually a process that the host uses to cover parasites that are endoparasites and sort of kill them or render them so that they can't reproduce and render them harmless. So encapsulation can be used on the animal, on things like worms. You can have what you see on the right there is a nematode or a little worm that's living in a host, and the host will actually cover it with specialized cells called lamellocytes. These are cells that insects produce, and they will actually cover the nematode and basically kill it. Encapsulation can also happen not just with the live animals, but with animal eggs, with parasite eggs. So what do parasites do? What do endoparasites do to resist encapsulation? A lot of them will produce specialized coatings. These they'll make cuticular lipids, these sort of fatty chemicals that prevent the lamellocytes from binding, and that basically allows them to get around or sneak through the process of encapsulation. Other parasites will use virus-like particles. They've actually sort of co-opted ancient viruses that are now part of their genome, and they'll use those viruses to inactivate or kill the lamellocytes. So again, it prevents the lamellocytes from binding, and it prevents that encapsulation process. Another thing that endoparasites have to deal with, like we said, is the immune system. And so a lot of endoparasites have specialized genes that allow them to get around the immune system. Some of them will actually change what the surface of their cells look like so that it tricks the immune system. The immune system will learn to lock on to one particular surface, and it'll develop all these memory cells to deal with that, and then the parasite will actually switch, and it'll put out different surface coatings, and that will cause it to look different, and the immune system gets confused. Another thing that parasites can do is they can sort of affect where they get moved into the body. And so malaria actually expresses proteins that allow it to migrate to the red blood cell surface. It expresses these proteins that migrate to the surface of the red blood cell, and that allows them to pull in nutrients into the cell and then it produces these knobs on the surface that cause the infected red blood cells to stick to the host cells and stick in that good environment where it's getting the nutrients it needs, and it prevents the malaria from traveling to the spleen where the malaria would otherwise be destroyed by the immune system. All right, so in the last class we talked about coevolution. We've talked about this in a couple classes before. It's basically that reciprocal evolutionary change in interacting species. In the last class where we were looking at plant-herbivore interactions, we saw that plants can evolve a toxin, and frequently the herbivore will develop some metabolic pathway to break down the toxin, or else it will evolve so that the toxin is no longer effective. And then the plant will evolve a new toxin, and we go round and around this cycle in the process of coevolution. Not surprisingly, coevolution also happens with parasites. So here I might have a plant parasite, maybe that's a rust, and it's got some particular sort of effector protein or some avirulence protein on its surface. And so what happens? Well, the plant might evolve a resistance gene. We talked about those plant resistance genes that are exactly the right shape, exactly the right puzzle piece to fit on and protect against that parasite. So there's my plant resistance gene, and it's active. It perfectly matches that parasite effector protein. And then what happens? Well, the parasite evolves to avoid that resistance gene. It evolves a new shape to those effector proteins, and now the plant resistance gene is no longer effective against the parasite, and so now it's susceptible again. So what happens? The plant evolves a new resistance gene. And so not surprisingly, this is why you have a lot of, when you have these gene for gene interactions, there can be many, many different variants of your plant resistance genes and your parasite effector genes because of this process of coevolution. The plant makes one resistance gene, the parasite makes a new effector gene. The plant makes a new resistance gene, the parasite makes a new effector gene. The plant makes a new resistance gene, the parasite makes a new effector gene. And so we get all of these different variants of these resistance genes and effector genes. And again, that's because the resistance gene is only active, it's only good against the one parasite effector protein. All right, there's a couple of different ways that genotypes can sweep through a population. So not surprisingly, selection favors certain genotypes. When we have a new parasite effector gene emerge through mutation, that can get around the resistance gene that most of the plants have. It'll go through and it'll become really the primary the primary effector gene in the parasite population because it's good at sneaking around the plant host until that plant evolves a way of getting around it. And so what this ultimately leads to are these dynamics. And remember, with population dynamics, we learned that dynamics is just change through time. And so you have these changes in the host and parasite genotype over time as a result of this tight interaction between the host trying to get around the parasite, trying to defend against the parasite, and the parasite trying to break through those defensive barriers. So there's a couple of different ways that these dynamics can occur. One is with recurrent selective sweeps. So this is where an allele provides an advantage. It's sort of an allele will emerge through mutation. It'll provide an advantage. It'll sweep through the population, become the most abundant. Most of the other alleles will sort of disappear, but then its advantage will be lost as soon as the other party evolves something that sort of defends against it. And so what you see here, maybe the blue is our host and the red is our parasite. And so the host will evolve some form of resistance, a resistance allele that's effective. It'll sweep through the population, but then the parasite will evolve some way around it, and that particular allele will sweep through the population, and then the host will evolve some new way of dealing with resistance for that new parasite, and that'll sweep through, and then we'll get these continual sweeps. And this is what we call arms race dynamics. We can also get what we call negative frequency dependent selection. This is where an allele provides an advantage, but only when it's rare. So we don't have that sweeping through and replacement. We don't have that new mutation continually evolving and replacing. Here we just have an up and down and up and down. So the host may have some beneficial allele that goes up, and it's beneficial until we have a parasite that sort of learns to deal with that particular allele, and that parasite will rise, and that'll cause that allele to go down, and a different host allele that is sort of provides resistance against the parasite that is now the most abundant, that'll go up until another parasite allele says, oh, well, wait, I'm good at dealing with this particular resistance form, and it'll go up, and we go up and down and up and down. And this is called rag-queen dynamics. Unfortunately, there is a cost to host and parasite defense. So both the host and the parasite, their counter-defenses in this coevolutionary process will get stronger and stronger and stronger over time. They might express more and more resistance proteins, and that can be costly. And so ultimately, these arms race dynamics are broken down once it becomes too costly for either the host or the parasite to continue in the game. It's basically a game of chicken. And both parties go up and up and up until one breaks. And so this ultimately leads to a trade-off where the defense or the counter-defense is too costly, and so it either lowers reproduction or it lowers survival enough that it's just not worth keeping that defense or that counter-defense, and one partner gives up. All right, unfortunately, parasites can drive host populations to extinction. Often this happens when we skip that coevolution step. If things are always co-evolving with each other, one is never really ahead of the other. Yes, one will give up, but usually at the point where they give up, neither is doing great. That's not true when we have invasive species. When we introduce new parasites, frequently we've skipped that coevolution process, and so the host is not at all ready to deal with the invasive parasite that we introduced. And often it can drive the entire population extinct. That's happened with a lot of different tree species: American chestnuts, oaks, elm trees, ash trees. That picture on the right is actually in the Smoky Mountains, really close to us. Those were the American chestnut trees, and now there are no large adult American chestnut trees in the Smoky Mountains. There are only small little baby plants that grow up until they get to a certain size, at which point the parasite hits them. Chestnut blight is the parasite. Not only can parasites cause extinction, they can also cause population cycles. So the parasite goes up in abundance, it causes the host population to crash, then there's not enough hosts anymore, and so the parasite population crashes and the host population goes up in abundance. Kind of like the lynx and the hare. It's exactly the same thing where one goes up when the other goes down. We can actually see this in this example. In this particular case, in the top left figure, that's the cycling grouse population. It's kind of bird, and it's cycling in response to a nematode or a worm parasite. What happened was in 1989, some grouse populations were treated for the worm. And what you'll notice is that in the figure in the top right, the oscillations are much less intense. They're more damped, and that's because we removed the parasite in 1989. In another population, that's that bottom figure, the parasite, the nematode, was treated for in both 1989 and 1993. And you'll see there's still some population cycles, but they're a lot shallower. It's a lot more damp. It's almost constant population size. And so what you see is that removing the parasite actually dampens the cycle. And that shows us that that parasite is driving the cycles in the grouse population. Not only does parasitism change population dynamics, parasitism actually changes species interactions. So parasitized individuals are weak, they're tired, they're struggling because their resources are being used by a parasite. And so even if the parasite doesn't kill them directly, parasitized individuals are much more likely to be predated on, and they're also much less likely to be able to catch prey. And so we see this in this example. Here we have tadpole mortality. Tadpole are basically little baby frogs. And what you'll see is that when the tadpole are infected, we have a lot more mortality than when the tadpole are uninfected. And in fact, without any salamanders, there's no mortality in the uninfected, and then there's some mortality in the infected. So the infection is probably causing some tadpole mortality either directly or by making it harder for the tadpoles to feed. But then what you'll notice is that when I put salamanders in the system, that's a predator, I get the salamanders eating some uninfected tadpole, but I get them eating tons and tons and tons of infected tadpole. And in fact, they're eating way more infected tadpole than they are uninfected tadpole. And so our mortality rate goes through the roof, not just because of directly this particular parasite, but being, this is a trematode infection, not just because of this trematode killing them directly or making it harder for them to get food, but because it's changing the amount of predation that's going on these tadpoles by the salamanders. Parasitization can also change competition between species. So frequently, without predation, one species will be a stronger predator and the other will be a weaker predator. But then when we introduce a parasite, we'll actually find that that reverses, and so the stronger competitor becomes the weaker predator when the parasite's around. And then parasitism can actually change whole communities, and it can change the physical environment. And I think this is a really great example just visually. So these are two beaches. And the difference in these beaches is that in one beach we have a trematode parasite that actually drives what we call an amphipod. It's like basically a little pill bug. It drives it extinct. And so when the trematode parasite drives the amphipod extinct, the amphipod is no longer around holding these mud flats together. And so that causes erosion, and it causes sort of that difference in beach structure. On the left, we have no parasite, and so we have the amphipod holding the mud together in those little clusters you see. And in the right, we have the parasite, so we wiped out the amphipod, and we don't have the mud being held together, and so we get erosion. So again, parasitism changes everything. It changes population dynamics, it changes species interactions, it changes, as a result of that, whole communities, and then it actually changes the physical environment. It changes the mud and sand in this particular case. And so everything can be altered by parasitism. So that's today's lecture. If you have any questions, I'd encourage you to shoot me an email. Otherwise, I will see you next time.